Hey.
Hey.
Where did you come from?
The talkative gene.
Oh.
Yeah. Mind if I express myself real quick?
Not right now. I'm kind of busy. I need to finish this.
It's crazy how the nucleus thinks that he's in
yesterday.
charge of everything when we're out here doing all the work. Like, I know he's the control center and the head of command, but I mean, he could cut us some slack.
Have you ever wanted to silence gene expression to stop unwanted proteins from expressing themselves? Now you can. How, you may ask? Let me tell you a story. One day in 1998, American geneticists Andrew Fire and Craig Mello wanted to see if they could stimulate muscle movement in some Seniorhabditis elegans by injecting them with messenger RNA that coded for muscle proteins. They tried injecting a single sense strand. Nothing happened. They tried again with an antisense strand. Still nothing. Then they injected double-stranded RNA, and the worms changed their behavior. They realized that by injecting double-stranded RNA, they could somehow silence virtually any protein-coding gene. So, what was going on? Well, a few years later, they got their answer. Allow me to introduce you to the concept of RNA interference. When double-stranded RNA is introduced into a cell, it gets recognized by RNA cleaving enzymes. The first one to take notice is DROSHA. It cleaves the RNA into a hairpin loop shape. This hairpin then gets transported into the cytoplasm, where another enzyme called dicer cleaves it further. The result are small interfering RNAs, or siRNAs. In the cytoplasm, a complex called the RNA-induced silencing complex, or RISC, is waiting for an siRNA. Inside this complex is another enzyme known as an Argonaute protein. Argonaute takes the more stable siRNA strand and incorporates it into the complex. Then the entire complex searches for protein-coding messenger RNA that matches the incorporated strand. Once it finds a match, RISC binds to the mRNA and Argonaute cleaves it, preventing more proteins from being translated and silencing gene expression. The good news is that if you miss your old talkative proteins, RNA interference is only a temporary silencing mechanism since it causes knockdown and not knockout of the gene. Since RNA interference silences genes, it has a lot of application in disease therapy. It's currently being used to stop viral infections and could potentially be used to halt the prognosis of cancer. RNA interference was a revolutionary discovery, and it's amazing how our own cells have such complex control mechanisms.